Hello students, in last class we have studied up to antigen. Okay, in previous classes we are discussing about acquired immunity and innate immunity. In acquired immunity, these are the things we are going to discuss in further classes. We have seen antigen. Antigen is nothing but it is a protein molecule that makes the immune system to response. Okay, now the antibody. What is meant by antibody? Who discovered? And structure of antibody we can see. What is meant by antibody? Antibodies are nothing but they are the proteins, okay, that can bind a antigens to form antigen antibody complex. Okay, where these antibodies are present? These antibodies are present in the blood cells. Once again I am repeating you students, antibodies are nothing but proteins which can bind with the antigens to form antigen antibody complex. We can see how it is going to bind, forming antigen antibody complex. It belongs to a group of immunoglobulin. Blood contains immunoglobulin, no? That the structure of immunoglobulin was discovered by discovery of antibodies was discovered by Gerald Maurice Edelman and Rodney Robert Potter. These are the two scientists who discovered antibodies. Antibodies are produced by immune system. How antibodies are produced? They are produced in immune system to respond to the entry of antigen into the body, which means that any foreign particle or antigen, when it is entering into our body, at that time immediately antibodies will stimulate and production of antibodies will start. All antibody molecules are immunoglobulin only, and chemically they are proteins. Usually we are to call as antibodies are nothing but proteins which contains a group immunoglobulin. We can see the structure, students. This is the structure of antibody molecule. Simply I will tell, just try to understand. If you see the structure, antibody is nothing but it is a protein molecule only, which was discovered by Porter and Edelman. If you see this structure, you will get into the mind, it is a Y-shaped structure. At the top it contains two antigen binding sites. Okay, na? It contains two chains. Okay, one is light chain and another one is heavy chain. In between disulfide bonds are present. It contains two variables, constant variable and variable region. Here we can look at the structure. Very important, students, very important. Definite question for 3 marks, either neat level diagram of antibody molecule or diagram with explanation, means 5 marks. If you see, this antibody molecule is made up of 4 polypeptide chains arranged in Y shape. They are arranged in Y shape, which contains 4 polypeptide chains. Okay, these are the 4 polypeptide chains, disulfide bonds. There are 2 long chains composed of 400 amino acids. Long chains contain this L chain contains 400 amino acids, and short chains 200 amino acids. Usually proteins contain amino acids, no? That one only. Long chains are called as heavy chains and short chains are called as light chains based on their molecular weight. Hence the antibody is represented as H2L2, heavy chains and long chains. All 4 chains are held together by disulfide bonds. Here we can see the disulfide bonds. They are held together. Variable region is the antigen binding region. If you see the variable region, here we indicates the variable region. Exactly at this region it contains antigen binding site that differs in different antibodies, whereas constant region is remain constant for all types of antibody molecules. Different classes of antibody produced in our body, like hemoglobin A, hemoglobin M, hemoglobin D, hemoglobin E and hemoglobin G, they perform their respective function. Hope you all followed this structure of antibody molecule. The important thing is diagram only. If you see the diagram, you can easily explain. It contains disulfide bonds, it contains antigen binding site, it contains 2 heavy chains, 2 short chains called as light chains. It is indicated as H2L2. Simply you can explain by your own words if you know the concept. Some extra information also given. If you want to know, just you can read it. It's not necessary. If you want more knowledge, means you can read, which means hemoglobin types, how they are functioning, that one they have given. Next, the difference between antibody, antigen and antibody. Antigen, immunogen, antibody, immunoglobin. Let's see differences. It stimulates the production of antibody. It is produced in response to antigen. With respect to antigen, antibodies are produced. What antigen will do? It is going to stimulate the antibodies. It may be protein, polysaccharide, lipid. Okay, na? It is a protein molecule only, antibody. It is variable in structure. It doesn't have definite structure. But antibodies have Y-shaped clear structure. We have seen the diagram now. Antibody diagram. Now 4th point is, antigen are nothing but foreign substance. Bring out reaction in the body. But antibody, it is a protective chemical. It is a protective chemical produced in the body. In the body itself it is produced, but it is a foreign substance which come from outside. But antibody not like that. It is produced inside chemically. Okay, na? And next concept is, students, acquired immunity is carried out by special types of lymphocytes. Lympho means blood, cytes means cells, blood cells. There are 2 types of blood cells: B cells, B lymphocytes, T lymphocytes. That mechanisms we need to study and the difference between that mechanisms. Just look at students, what exactly means lymphocytes. Lymphocytes are the WBC cells originate from the bone marrow. How they are going to originate? They are going to originate from the bone marrow. Our body contains bone marrow, no? Exactly from that bone marrow, the cells are going to originate. When they originate 100%, about 50% of lymphocytes reach the thymus gland. Thymus gland is nothing but it is present exactly at the breast region. That is a small organ or bone. It is going to call thymus gland or breast bone, and differentiate into T lymphocytes or T cells. Remaining 50% of lymphocytes do not reach the thymus gland but differentiated into B lymphocytes or B cells. In addition to B cells and T cells, there is another group of lymphocytes called as natural killer cells. Here we need to study the mechanism of B lymphocytes as well as T lymphocytes. Just look at this, try to understand. It is very easy concept when you study. B lymphocytes, now we can see antibody mediated immunity, shortly called as AMI, or role of B lymphocytes, or humoral immunity. Humoral immunity is going to call. B lymphocytes acts indirectly against antigens by producing antibodies. What B lymphocytes will do means they are going to produce antibodies to antigens. This is called humoral immunity. Humoral immunity means B lymphocytes acts indirectly, not directly, against antigens by producing antibodies. Next, the antibodies are carried by the body fluids like blood and lymph and react in various ways to destroy specific antigens, which means that the concept is going to tell in our blood cells, okay, that is B lymphocytes and T lymphocytes, in blood itself the antibodies are produced to specific antigens. Next, when antigens enter the tissue, B cells are stimulated to produce antibodies. See, when antigens are entered means B lymphocytes stimulate antibodies. There are thousands of antigen specific B cells present in the body. Thousands of antigens are present in the body. There are thousands of antigens specific B cells present in the body already, which is present in the body. The membrane of each type of B cells can synthesized by the contact of an antigen. Once an antigen specific B cells is, when it is activated, means it multiplies very fast to produce a clone of plasma. Plasma cells or blood cells, both are same. Once an antigen specific B cells is activated means it multiplies very fast the plasma cells. These plasma cells produce antibodies 2000 molecules per second. These antibodies combine with the antigen to form antigen antibody complex. Once this antigen antibody complex is formed means then fighting will start, which means that to antibody it contains antigen antibody complex like this. Like this antigen binding site. When it binds means fighting is going to start and killing that antigen. That's the thing it here told. These antibodies combine with antigen to form antigen antibody complexes. These complexes are going to enhance these type of phagocytosis, vasodilatation, vascular permeability to proteins, neutrophil exudation, these things they are going to enhancing. Next, cell mediated immunity or role of T lymphocytes. It's shortly called as cellular immunity. Here T lymphocytes acts directly against antigens by attaching themselves to the antigen powering cells, that is cell to cell contact is called cellular immunity. Cellular immunity means there will be cell to cell contact. Okay, na? With the help of cell, the immunity is developed and killing that antibodies. There are about thousands of different types of T cells. Each one is capable of responding to a specific antigen. If you see the cells, there are different types. There are different types of T cells. When an antigen enters the body, a particular type of T cells which is specially programmed to react with the antigen become activated and synthesized. These cells increase in size and multiply rapidly to produce clone of T cells. What are the different cells we can see and their function? Killer T cells, helper T cells, suppressor, memory T cells. Killer T cells. These cells migrate to the site of infection and attach to the invading microbes or antigen and secrete toxic substance, cytotoxins, and destroy the microbe directly. Helper T cells. These cells stimulate B cells to produce antibodies which interact with the antigens of the microbes. Next, suppressor T cells. These cells suppress the immune system after the antigen or microbe is destroyed. It is going to suppress the antigen. Memory T cells. These cells are pre-programmed to recognize and respond very quickly against an antigen that may appear in the future. Memory T cells function is it is going to recognize the antigen when an infection occurs once again. It is going to recognize and killing that antigen. Okay, last thing we can see, students, the difference B cells and T cells. B cells are produced by bone marrow. This is also produced by bone marrow. But in B cells, they differentiate to bone marrow itself. But here they differentiate into thymus gland. Thymus gland is nothing but it is a small organ which is present behind the breastbone. Okay, na? That is responsible for immune system to produce. On exposure to antigen, become active. B cells, they become active when they are exposure, multiply to produce clone of plasma cells. Here plasma cells are produced which secrete antibodies. Here on exposure to specific antigen, become active, multiply and produce different types of T cells. Here plasma cells produced, here T cells produced. Remaining thing is same, activating of antibodies. Next, B cells attack antigens directly by producing antibodies. Here T cells directly attack antigens. Here indirect, here direct. They secrete specific antibodies. They secrete, T cells secrete cytotoxins and other chemicals to destroy pathogen. B cells have short lifespan, it have long lifespan. Okay students, this is about acquired immunity which is carried by B cells and T cells or B lymphocytes or T lymphocytes. Hope you all understood. If you have any doubt regarding this concept, please drop a message. I will clarify your doubt. Okay, now try to understand the concept, listen the video carefully and make points which will help you for your examination. Okay, thank you. Remaining class continue in tomorrow.